Hello, everyone, and welcome back to class. I'm really excited to meet with you once again and show you a new and awesome biological concept. And that is going to help you further understanding in theme 2 of the class, where we talk about the smallest units of life and how biologists study those small units to be able to inform themselves on more complex ideas and concepts that we will, of course, encounter later in the course. So this week, we are going to segue and scaffold off of our previous week. In our previous week, we learned about biomolecules, and we use that as a platform or foundation for this week. Just to reiterate what biomolecules are, biomolecules is essentially the fuel that our cells need to be able to survive and maintain one of the many characteristics of life and maintain homeostasis or cellular stability. Our bodies are full of cells, and we are one organism among many within a broader group called the eukaryotes. And eukaryotes are identified based upon their cell structure. And we can take a look at that cell structure this week by looking at the characteristics or general morphology or shape of those cells. Because if we take the eukaryotes, we can actually use other characteristics and be able to split that eukaryote from plants, such as that possess a cell wall, to animals, which do not possess the cell wall and have a little bit more complexity to them. On the other branches of the family tree of life, we can take a look at the prokaryotes, which is our other group that we need to be able to understand and acknowledge that they exist. Prokaryotes are divided into, let's say, younger and older groups. The older groups are going to be like early bacteria. They're called archaea. And these are very restricted in where they live. They tend to live in really extreme environments because they're adapted to be able to live only in those environments. And then we also have the bacteria, which are single cell prokaryotes. So this week, the general theme is, does it have one cell or does it have many? Is it more basal or basic, or is it complex? And that's what we're going to do, is we're going to investigate and take a look at the morphology of different cells. And so now I'm actually going to show you a little bit about this week's lab assignment, as it is going to be one of the more interesting ones because you are actually going to be using another open educational resource. And that open educational resource, or OER, is going to be a virtual microscope, a virtual light microscope, typically one that you will use in many science classes in person. But seeing that we're in an online learning environment, I am bringing the tools to you. This virtual microscope is produced by the North Carolina College Bio Network, and they have a lot of really great resources that can help you further and master the content in this course, but we're really just going to be using that virtual microscope. All right, so I just clicked on the link from our Brightspace launch activity. And so you'll just have to give it a couple moments to load, but this is a really cool resource, as it's a recreated 3D workspace for being able to use a virtual microscope. You can click on the icons below. They actually have a guide that gives you some really good facts and information that I recommend you investigate, as some of your questions actually come from the text in this material. How objective lenses work and how to select them. Difference between immersion oil lenses and regular ones. how to even care for a microscope, just in case you want to go and get your own microscope one day. And vocabulary. But the real fun is going to actually be able to explore this microscope. You will be able to go and take a look at virtual slides. So one of the things you're going to be doing in this lab is you are going to investigate the two broad types that I want you to learn a little bit more about in your lab. You are going to take a look at slides dealing with prokaryotes and eukaryotes. So what you can do is click on Explore, and then it brings your microscope up, and then you can take a look at the different slides. So let's actually just familiarize ourselves with how to use this virtual microscope. We can take the letter E, and we can change our focus using the sliders at the bottom and adjust our lighting. So that way we can see all the detail in our slide. And so let's take a look quickly at some bacteria, acid-fast mix. So yeah, take a look at some of that bacteria. We can even go to a oil immersion slide, add a little bit of oil to it. Oh yeah, take a look at all that bacteria in there. I think that's pretty cool. And so that will represent your prokaryotes. And then you got to clean up your microscope. And then let's actually take a look at some animal and plants. So I'm looking at our directions in our lab. So let's grab some plant cells. And there you go. You can easily identify this eukaryote being a plant cell because it has some nice, wonderful cell walls. The little green discs are going to be your chlorophyll. You got a nice centrally placed nuclei, which is diagnostic for the eukaryotes. And then we can take a look at some human. Let's take a look at... Now let's take a look at some simple squamous epithelium. This is actually taken from the kidney. Add a little bit more immersion oil. You can actually move the slide around a little bit, but you can see some really good characteristics of an eukaryote cell. I mean, that dark nucleus, it's got a relatively large size compared to the prokaryotes we saw earlier. And a loosely bound cellular membrane. Remember to clean up and put your slide back. And so that's really what you're going to be doing. You're going to go and you're going to investigate. You're going to take screenshots and then document those screenshots, because that's the thing about lab is I want you to be detailed in your descriptions, but I also want you to document things as much as possible. If you got any questions, I'm happy to meet with you during open office hours through email, video chat, or just swing by my office. I've got a lovely selection of snacks, and you're more than welcome to swing on by. All right, let's get learning.